start this week talking about enzymes and we'll end the week talking about cells. I just wanted to give you a little rundown of the PowerPoint I'm going to use to introduce enzymes with you. Enzymes, whoops, sorry, clicked the wrong thing. Enzymes are pretty important for us because they are proteins, which is one of our molecules that we have talked about last week as being one of our important macromolecules. And they are important as far as making sure reactions occur in our bodies. So when we digest our food, starting in our mouth with our saliva, these guys are carrying out that process. We swallow our food and we have acid in our stomach as well as enzymes, which are proteins that act as catalysts, which speed up the chemical reactions of digestion. So here you have a very simple picture, and this picture is labeled later in the slideshow. This orange blob is the enzyme. Notice it doesn't change. The enzyme has this red and green dot. If you're colorblind, I apologize that it's red and green. Comes into the substrate here, and then it releases as a purple dot. This is called synthesis. It starts off as two separate dots, comes together, and we form a purple product. These are reactants. This is the substrate. These little grooves here are the active site. This middle section is the enzyme-substrate complex. This is still the enzyme. Now this is a product. So what are enzymes? Enzymes are basically catalysts, which are proteins, the macromolecule proteins. So they're made as long chains of amino acids, just like we talked about in our last section. But they're catalysts which help us make reactions happen. We're all frugal at some point. We want to get the most bang for our buck. We want to save money. So we want to spend less and get the most, and that's what our body wants to do too. So we want to get the most for our money, and so our body wants to make the most happen for the little cost. And that's what these guys are here. These enzymes down here, they're making the most happen at least amount of cost. Enzymes are recyclable, so they're not damaged unless heat, pH, or salinity is in the picture, which we'll talk about in a second. So these are some important points for you to note about enzymes. They're proteins. They're folded into specific shapes. Like I know most of you know my hair is really curly because it has extra protein in it that makes it super curly. If I heat it and I straighten it, I break those proteins and I make my hair straight. So that's important to note. I can chemically straighten it. You can chemically curly straight hair. Those things are adding proteins and taking proteins out. So that's just a real world example of how proteins work. Enzymes are there as things that help us react. So again, here's a enzyme. Here's a substrate. The substrate fits into the enzyme at what's known as the active site. That's this little groove right here. It's pictured here and labeled for you. Notice something happens to the substrate. It's going to digest in this example. The first slide, it was forming, it was synthesizing. And as I digest, I release these products, this product here. So it starts, if you remember in chemistry or physical science, you have reactants on the left-hand side of the equation. You have products on the right-hand side of the equation. Same idea here. You have reactants on the left, products on the right, but we're just lowering the energy required for this reaction to take place because we have an enzyme present. There is an animation I want you to watch. It's about a minute long and it shows you this thing in real life, and I just want you to watch that. I don't want to bore you with that at this point in this slideshow, but I want you to watch this once it's shared with you. Okay, so enzymes are there to catalyze reactions. Here's some more points about them. They're reusable, which means they don't get used up. They usually end in ase, and some of your vocabulary words. Sucrase is going to break down the sugar sucrose. Lactase, if you're lactose intolerant, that means you don't have lactase in your stomach. And then maltase is going to break down the sugar that's found in things like beer, things that are involved in the alcoholic brewing process. So notice these end in ase. If you put an ose at the end of these, those are going to be the sugars that they break down. Again, there's your substrate, there's your active site, and this green blob would be your enzyme. Now, how do enzymes work? They're just going to lower the energy necessary to push this little marble over, or this little ball over the heel. So instead of the heel being steep and there being a lot of energy used, we're just going to have to go over this little tiny little blimp. It's not going to be this huge mountain. So instead of us going way up here and way down, we just have to go over this tiny little hill. So not near the energy necessary, because there's a place for the reaction to take place. So when an enzyme, the reaction is going to happen regardless. Let's clear this up. Whether an enzyme's present or not, the reaction will take place. But an enzyme's going to let it happen cheaper. Think about it financially. If I want to get the most bang for my buck, I'm going to go to the sale rack. So enzymes are like the sale rack at a store. I'm going to get the most bang for my buck because I have an enzyme present that's going to let this reaction proceed. So here's my reactants. This is like having H2O, water. And then I'm going to let my reaction proceed into hydrogen and oxygen down here. It's not the best example, but you get the idea. Sodium chloride, salt. My reaction proceeds and I get sodium and chlorine. It's all decomposition I'm talking about here. But at any rate, the reaction happens at a lesser energy cost to the cell and to the body because there's an enzyme present. It's all about saving for the body. Another example here, the reactants, that's what you start with. The blue line is with an enzyme, little baby heel. The red line is without an enzyme, a great big ginormous heel right here, and then the products are down here at the bottom. Notice that activation energy, which is the energy required to get the reaction started, is just a little bit when you have an enzyme, but it's a huge amount when you don't have an enzyme. That's what your body wants to avoid, this right here, because there's too much energy required when you don't have an enzyme just to get the energy, just to get the reaction started. So we want to avoid this. We want this. So if there's an enzyme present, the reaction is going to happen at that location. So again, here's an enzyme-substrate complex. That just means that the substance, which is the substrate, or the reactant, is going to act on the enzyme. So the substrate is what starts at the left-hand side of a chemical equation. It's going to go in and fit perfectly like a lock and key. So this substrate is going to fit perfectly into this enzyme. If I took the key to my house and tried to go into your house, it would not work. It doesn't fit perfectly there, so that's important to know. So substrates and enzymes fit like a lock and key. The active site, that's this location, perfectly where it fits. If it doesn't fit, my key may go into your lock. It's not going to open your door, though. So that's got to be a perfect fit, a hand in a glove. It's got to fit perfectly. And that doesn't look like a perfect fit, but it does. Induced fit means sometimes that the enzyme and the substrate, once the substrate gets in there, it may wiggle it around to make the, let me back up. It may change that substrate just a little bit to make it fit. Induced fit just means that it's going to force it to fit. Now, one last thing. There's 3 things that can change the way. Well, one thing I'm going to focus on that can change the way an enzyme acts, and that's environmental conditions. We're not going to focus on cofactors or enzyme inhibitors. So let's look at these environmental conditions. One can be temperature. If you heat something up enough, it's going to morph the shape or change the shape. So if we have an extreme temperature like a high temperature, it's going to do something called denature, which means unfold the enzyme. When that happens, it's inactive. It's not going to work. pH. If we change, most of these are occurring in our body. Guys, your body's pH is around a 7. So if we're around a 6 to an 8, we're good. But if for some reason we're sick and our body's pH changes, which can happen, then your enzymes aren't going to function properly. So you're going to have some denaturing of those enzymes and you're going to get sick from that. And then salty, salinity, which is often called ionic concentration or salinity. And that means that, no pun intended, but you're salty. And if that happens, then your enzymes aren't working properly. So these 3 things can change the way an enzyme works. Temperature, mostly high temperatures, pH, and then salinity. Now there's 2 videos and I'm going to have a worksheet that goes with the Amoeba Sisters video that you'll watch today, Monday. The Bozeman video is just an additional video, and then there's a textbook link here that I've added that will take you directly to the section. You might have to scroll down just a little bit, but if you have questions, you can also read a section in your textbook that has to do with enzymes. We're going to spend today, tomorrow, and part of Wednesday on enzymes before moving into prokaryotic and eukaryotic cells. Hope y'all have a great day. See you soon, hopefully. Oh, and here's one last picture, sorry, of an actual enzymatic reaction. You can look through this as well.